purpose is to disseminate knowledge, increase overall knowledge pool of this society. And in this series, today's speaker is Dr. Hina Vadera. She has recently completed her PhD from Bhavnagar University on source sink relation, and she is recently teaching at Sir PP Institute of Science, Bhavnagar. She has also published a research paper. I'm not audible. Am I audible?
Yes, sir.
Hina, please check your internet. I'm audible. So I welcome Dr. Hina in this Science Dialogue series.
Thank you, sir.
Now platform is yours, and this is request to the participants that attend complete lecture, and you can also interact if you find something difficult during lecture. But generally we allow interaction after lecture. But if you find something very necessary, then you can unmute and speak and ask question. And after lecture, we share Google form, feedback form. If you need e-certificate, then you can fill your feedback so that you will get e-certificate also as a participants. Don't ask me e-certificate after this lecture or this link. So please attend complete lecture and interact. You will definitely enjoy. And this topic is very relevant for graduate student, postgraduate student. So I request you all to attend. Now, so we are thankful, Hina, for joining us and for accepting our invitation to deliver lecture. Welcome. Now,
Thank you,
platform
sir.
is yours.
Thank you, sir.
Please, now you can start.
Yes, sir. Good evening to all. As sir introduced me, I am Hima Vadera, visiting faculty from Botany department, and today we will discuss about plant tissue culture. We all know, or we all a little something this area, but how it will affect our economy, how it will affect our plants, and how it affect will our medicines and food also. We will discuss the primary outline of what is plant tissue culture and what where it is. This is the outline of the lecture. What is plant tissue culture? The primary history of it, basics of plant tissue culture, laboratory organizations, nutrition medium, stages of plant tissue cultures, types of cultures, application and its limitations. Firstly, what is tissue culture? Tissue means the mass of cells which is used to culture whole plant, cells, tissue or organs in a sterile artificial environment in controlled nutritional and environmental conditions. It is not a subject we study as a subject, but it is a part of range of techniques in which the knowledge of many different scientific disciplines are combined and used. It is widely used in world as well as our country also, and have a large applications in modern biotechnology. We move to the history of plant tissue culture. German scientist Gottlieb Haberland, known as a father of plant tissue culture techniques. His work on the single cell culture paved the road for the subsequent development of plant tissue culture. While in 1962, the major contribution of Murasich and Skoog is to development of nutrition medium for the plant tissue culture. While Indian scientists such as Maheshwari and Guha successfully cultured the haploid embryo with the help of pollen grains of datura. This is the pioneers of plant tissue culture in world or in our country also. If we know, or if we want to know actually plant tissue culture is, we have to know about their basics, such as we have to know that plants are the only organisms having a totipotency. Each cell of plant have totipotency, which is the ability of cell to differentiate and develop into a whole plant when given suitable condition. It is not possible for human except stem cells. But if we need or if we want to culture a human with a cell of finger or cheek cells, we cannot do that. But in plant, it is possible due to that totipotency. We can see that with the simple example of carrot plant, the cells of root transfer to the growth medium and it starts divide and differentiate and can form a young plant or adult plant. So move to the other basics such as dedifferentiation, redifferentiation and callus. It is a capacity of differentiated cells to become an undifferentiated mass of cells, which means if the plant cells such as parenchymatous, sclerenchymatous and collenchymatous, which is assigned its confirm work, but we can turn into undifferentiated mass of cells, and the ability of cell is known as dedifferentiation. It helps to restore meristematic condition in the tissue, and the further development of a new young plant is possible by redifferentiation of the undifferentiated mass of cells. The mass of undifferentiated cells known as a callus. Move to the explant. This is the necessary part of the plant tissue culture because without explant we cannot culture a new plant, new cell or new organs. So explant is a living tissue which we collect from the desired plant and transfer to the artificial medium for the culture. In short, the explant is the small sterile piece of plant tissue such as leaf, stem, root, embryo, flower, etc., taken from the parent plant and used as a starting material to regenerate the whole new plant in a laboratory. So, another necessary requirement is the aseptic condition because if if we know what is plant tissue culture, everyone know that explant is just like a pampered child. You know, if there is a such difficulty, it cannot survive because it is far from its natural habitat. It is grown in a laboratory in vitro setup. So we have to maintain aseptic condition. Plant tissues grown in vitro need a nutrition rich media because we don't provide its soil. So we have to give its media, which is also an ideal breeding condition for the growth of microorganism. And without aseptic condition, the microbes can quickly grow and destroy the plant tissue and make its own colony on the nutrition media. So aseptic condition in plant tissue culture involve creating and maintaining a sterile environment to prevent contamination from microorganisms such as bacteria, fungi, and viruses. And to achieve this sterilization, there are many methods, sterilization methods, including autoclaving, dry heat, chemical treatments, ultrafiltration for media, as well as equipment. Coupled with the strict operational protocols, one should maintain strict protocol to maintain this aseptic condition. One should wear a protective gear like mask, gloves, ensure the integrity and success of the tissue culture. Further, we have vast and full equipped laboratory organization including washing area or facilities, media preparation room, transfer room, culture facilities and greenhouses. This is the ideal plan for plant tissue culture laboratory. We can see in corner there is a media preparation and sterilization room, followed by washing room. Media preparation room should have the media itself because the chemicals, lab benches, autoclave, weighing balance, pH meter, etc. Because this instrument will help to prepare a successful media for the tissue culture. While washing room will have basins, running tap water, drainage system, racks, etc. for the glassware washing as well as explant washing, and there should be a dry heat oven or dry air oven to dry the glasswares. On the other hand, storage room contains the racks which store the chemicals, glasswares, and prepared media. Dark room is required for the storage of samples of plants as well as some chemicals which need to away from the light, and refrigerators, deep freezers for the sample preservation, and transfer room is used for the inoculation of explant into nutrition media, and transfer room contain laminar airflow with HEPA filter, and hello, am I audible? Hello.
Yes, yes, you are audible,
Okay, okay,
but you
sir.
are not visible. कैमरा ऑन कर लीजिए अगर कर सकते तो.
Okay, sir.
Yes, please continue. Slide दिखा.
Okay. So in transfer room, the inoculation will take place, and for that we have the laminar airflow, which is a hood, having a hood and UV lamps and HEPA filter for the sterile transfer of explant into the nutrition media. In with this, there should be a main room having a benches, tables, microscopes, stereo microscope, cell counters, which we will used for the preparation and identification of cells, some chemicals, and etc. Culture room contains some racks which we will use for the to put our culture tubes, and also the light controlling units and tubes for the temperature control and humidity control. After that, greenhouse are there. Greenhouse are used for the acclimatization of plant, because if we grow the plant in vitro, it is not susceptible directly in the field. So we have to make sure that it is acclimatized to the environment firstly and then transferred to the field. For the acclimatization, we will transfer our cultured plant into greenhouses. These are some instrument we used in a plant tissue culture, or these are the essence of tissue culture lab. Weighing balance lab, weighing balance are used for the media preparation. pH meter is the must because we have to make favorable pH of each nutrition media with it varies with the plant of the species, and hot plate with stirrer, heating mantle, distillation unit for distilled water, hot air oven for drying glasswares, laminar airflow, and autoclave. Laminar airflow is the must for the plant tissue culture because of it is protect culture from dust particles as well as microbes, while autoclave is the pressure and temperature mediated sterilizing unit which we can use for sterilization of media and glasswares. This is the culture room, how it maintained with the light, which is adjustable as per plant's need, and temperature controllers, and there are racks, and there are the tubes which or jar which we used for the culture. We move to the nutrition medium. Nutrition media consist nearly similar composition found in natural soil, because we will grow it in lab, but it requires same composition of inorganic salts, carbon and energy source, growth regulators, vitamins and water. This is the standard composition of nutrition media given by Murashij and Skoog, and it consists macronutrients, micronutrients, and firstly we make their stock solution. Stock solution we will make stock solution of macronutrients, micronutrients, vitamins, and other freshly added compounds are there. We can say that macronutrients nutrient consist calcium, magnesium, potassium, phosphorus, iron, and chelating agents like EDTA. On the other hand, micronutrients such as boron, manganese, zinc, iodine, sodium, molybdenum, copper, and cobalt, along with chlorine added to the stock solution of micronutrients. Each stock solution mixed and in some amount, and we will prepare the working solution, and their concentration are given in the last column. Similarly, vitamins such as nicotinic acid, thiamine, pyroxidine, and glycine were used as a source of secondary metabolites sometimes. Growth regulators. Two hormones affect plant differentiation. First is auxin and second is cytokinin. These are two major affected growth regulators of the plant tissue culture. Auxin stimulates the root development, while cytokinin stimulates the shoot development during the plant tissue culture. Generally, the ratio of these two hormones can determine the plant development or tissue development. Higher auxin and lower cytokinin content initiate the root development in the tissue, while higher cytokinin and lower auxin concentration induce shoot development. The similar ratio or their maintained ratio induce the callus development in the plant tissue culture. These are the stages of tissue culture. In first stage, selection of healthy parent plant with desired characters will be done. We have to select healthy plant for the plant tissue culture. If the plant is contaminated or infected, it will ruin the whole protocol and whole process. So selection of healthy parent plant is must. It also includes the sterilization of explant tissue. After selection, collection of explant will be done, and sterilization of explant tissue will be done with the special chemicals such as sodium hypochlorite, mercury chloride, and sometimes if there is a lower chance of infection, we will use the detergents such as T-pol, and their concentration may vary with the how what is the explant, if leaf, if stem, if hard tissue, it vary with the concentration of sterilization process or compound, it will vary with it. In second stage, inoculation and induction will be done. The sterile explant tissue were transferred to the growth medium. This process is known as inoculation, and there are growth medium or growth regulators found in the growth medium. It will induce the tissue culture. So, in for a second stage, inoculation and induction will take place. Explant placed into growth media. In third stage, subculturing and proliferation. What is subculturing? Once explant transferred to the growing media, it will start to divide and differentiated, and that differentiated cells are then cut into small pieces and transferred to the other media for rooting or shooting or whatever we need from that cells. So subculturing and proliferation is the process that explant transfer to the shooting media or rooting media. Then after that rooting stage or stage 3 around, in this stage in vitro regeneration of shoots transfer to the rooting media. So after the shooting media, it transferred to the rooting media. In general, in general terms, during the cell culture, so a cell will be divided in the callus transferred to the shooting media, and shooting after the shoot formation occurs, it was transferred to the rooting media for the generation of root. So these are the general protocol or general stages of tissue culture. In last stage, hardening and field transfer. As we discussed before, plantlets are susceptible to infection if directly we transfer it to the field. So firstly, we were applied to the hardening. In this, the plantlets transferred to the mist chamber or glass house for the hardening and acclimatized to the open environment. Fully acclimatized plants are transferred to the fields. The maintenance of culture is necessary. Without this, your experiment will fail. If you not maintain your culture, it will not helpful. So there should be culture room or growth room. Proper photoperiodic racks. Photoperiodic means each plant have specific need of light, specific need of temperature, specific need of humidity. So, photoperiodic racks are the racks which can maintained by controllers, and with the requirement of plant, it can change the timing of photo, so light. So there is a range of 16 years to 8 years photo period. Temperature maintained, there is a 25 plus or minus 2 degree temperature can be maintained in the culture room or growth room. Now, types of culture. Organogenesis, it self-explanatory. Genesis means to generate an organ. So if with this type of culture we can form a organ. And second one is somatic embryogenesis. Third one is embryo culture, haploid plant production, and protoplast culture. These are the, I have taken these main types of culture. There are many subtypes, cell culture, cell suspension culture, but these are the major types of the plant tissue culture. Firstly, we study about the organogenesis. In this, the explant transferred to the growth media, and it will, if it will direct form organ or it will form callus. On the basis of that, it is further divided in two types: direct organogenesis and indirect organogenesis. Direct organogenesis is the direct organ formation from explant without callus formation. There is no formation of callus during this process. We will say that if we transferred a explant from leaf to the growth media and apply higher auxin or higher cytokinin ratio to the explant, it will form a shoot or root. So there is no formation of callus will happen. And if we will manage the auxin cytokinin ratio, so it will be formation of callus there. So that type of organogenesis is known as indirect organogenesis. We can see in this diagram, this is the explant. From this, we we can direct form a root or shoot in the plant. While in the indirect organogenesis, there is a formation of callus from the explant, and this callus then treated with the proper and if proper growth regulators and form a shoot and root through the callus formation. So this is the two types of organogenesis: direct organogenesis and indirect organogenesis. Then somatic embryogenesis. Somatic means cells of body or cells which not responsible the synthesis of generic cells or reproductive cells. And embryogenesis is the formation of embryo. This is the production of embryo from the somatic or non-germ cells. In this process, explant will turn into group of meristematic cells, generally known as callus, and these cells are transferred to the somatic embryos. Especially this technique known or used for the artificial seed production. We can see the first image showing the callus, while the second image showing the somatic embryo, which is formed from the somatic cells. It is the general protocol in which the cell of explant will be placed in the growth medium, which divided in a cell clump or calens, and in first medium the initiation of embryogenesis. In this process, the cell transferred or cell will treated through the growth medium to prepare as a embryo. So these are the induced cells, and in second medium it will form a embryo like structures, but it is not a proper embryo, so it will transfer to the medium having a abscisic acid, which is responsible the formation of embryogenesis in the normal condition. So it will apply to the that medium, and then it will start to develop globular stage, heart stage, and torpedo stage, which we found in a normal embryogenesis, and it will finally create a plantlet. So this is the somatic embryogenesis. After that, embryo culture. Explant, as an explant, we will take the embryo of the plant. Why embryo? Because some plant or some hybrid plants do not develop the embryo during its life cycle, and then there is a no chance of new plant generation there. So at that time, we can take embryo as an explant and rescue and overcoming dormancy for it. And this is their protocol. This is the example of Hordeum vulgare and Hordeum bulbosum. This is the hybrid ovule, and after surface sterilization, we will cut the micropial end and put the hybrid embryo in the seed. After that, the embryo transplantation in the endosperm, and after that, whole endosperm was inoculated in the culture dish. And after the culturation for some days, we can found a seedling. So it is useful for the embryo rescue in especially rare hybrids. Haploid plant production. In this, anther culture or pollen culture, with the help of that, and ovary or ovule culture, we can forming a haploid plant. production. Especially culturing of anthers or pollen grains derive a mature plant from a single microspores, and culturing of ovary or unfertilized ovules form a single mature plant. So this is the protocol of anther culture. We have to take unopened flower buds with surface sterilization. We have to dissect stamens. There are many stamens, but we have to check what are it that is suitable or viable. So we have to taste through acetocarmine squash test, and suitable anther will transfer to the nutrition media. And after that, the nutrition media, the embryogenesis will start, and that they were transferred to the rooting medium, and new plant will grow as a haploid plant. Now, protoplast culture. Protoplast culture is the useful for the plant tissue culture. It involves isolating and culturing naked plant cells. Firstly, what is protoplast? Protoplast is the cell without cell wall. It includes cytoplasm, cell membrane, and nucleus. So, protoplast is the culture of naked plant cell. Naked means having no cell wall, which is to generate entire plants. This method is used for the plant regeneration and the creation of somatic hybrids. Somatic hybridization includes the hybridization of plants without the involvement of germ cells. So, with the help of protoplast culture, somatic hybridization will take place, and the direct introduction of foreign genetic material via transformation techniques can be done with the protoplast culture. This is the protocol of protoplast culture. Firstly, we have to take cells from the regenerated plant, and with the help of centrifugation, we have to remove cell wall from the cells, and the protoplast will the found in the supernatant, and then isolated protoplast will plating on the plate and induce the wall generation. After the first division, we found callus, and this clump of cells made a regenerated plant. There is a two method for cell wall removal. One is the chemical method, and second one is mechanical method. In chemical method, we will use pectinase and enzyme, cellulase enzyme to degenerate the cell wall. In mechanical uh, method, we will crush the plant tissue and then centrifuge the plant tissue. There will be two types of isolation. Factor affecting tissue culture. The in vitro growth and development of a plant is determined by several factors. The genetic makeup of the plant, source of explant, nutrition, environmental conditions, including light, temperature, pH, oxygen, and carbon dioxide concentration. The genetic makeup of plant is very crucial for the plant tissue culture, because on the basis of that only we can imply other three factors to the tissue culture. Source of explant. If we talk about the explant, whether it is young or old, usually the younger or less differentiated explant have the more more result and the better for the tissue culture. On the other hand, type of explants such as leaf, stem, root, meristem also affect the growth medium, their growth ratio. We move to the growth medium, which is fully artificial. We are making them in vitro as a soil. So nutrition such as macroelements, microelements, plant hormones, vitamins also affect the tissue culture, and each species have the unique concentration for the growth. So we have to standardize the protocol according to which plant we want to grow through tissue culture. Environmental factors such as light intensity, as we discussed before, photo periods also affect temperature, humidity also affect the tissue culture. Now we move to the application of plant tissue culture. I believe this chart will help to understand how plant tissue culture have how many application it have. First is micropropagation, cryopreservation, extraction of secondary metabolites, crop improvement. Micropropagation is the method with the help of a small explant we can prepare or produce many plants or they having identical copies. So it is useful in the production of horticulture crops, agriculture crops, rare plant species. If there is only one plant in the country or specific one or two plant found, that is endangered or rare species. So their hybridization or other with the other techniques, it is not be produced more. So with the tissue culture, with the help of one leaf, we can make 100 or 1,000 copy of rare plant species. So this is the power of tissue culture or power of micropropagation. So it is very useful for that. And sorry, many government scientific research centers, it promote the tissue culture for the rescuing the rare plants. Third one is like agriculture crops, we already discussed. Plantation crops plus medicinal crops. With the help of tissue culture, we can grow more medicinal plant to overcome the requirement of large population. So it is the micropropagation application. Second one is cryopreservation. With the help of plant tissue culture, we can develop a plant from the germplasm storage and DNA libraries. With the help of them and biotechnology, we can produce desirable characters, plant with the desirable characters. Third one is extraction of metabolites. Extraction of secondary metabolites. Metabolites, each organism have two type of metabolites. Firstly is primary metabolites and second one is secondary metabolites. Secondary metabolites are the metabolites without them plant can survive, but with the help of them they have such specific abilities. If we talk about the tulsi basil plant, there is a higher amount of aromatic compounds and phenols which are secondary metabolites of the plant, and with the help of that we can have their medicinal value also. So with the help of tissue culture we can manipulate the cells producing these secondary metabolites, and through that we can get our antibiotics, alkaloids, hormones, pesticides, and medicinal uh, chemicals. So, through the plant tissue culture, we can form the major factory, natural factory to formation of some compounds are used for us. And last one is crop improvement. With the help of plant tissue culture, we can grow the disease resistant plants, drought resistant, high yielding or high quality plants, and short season yield. We know that some plants, if we talk about mango plant, if we plant a seed of mango, it will take up to 15 years to form a fruit. But if we try to culture the plant, so we can shorten the cycle, shorten the season cycle, and have a large population, large production and early production. So it will help to short season yield, reduction in fertilizer application. If we consider, if we success, if we can grow plant with desired characters, healthy plants, so there is no need to use fertilizers. Through tissue culture, we can prepare the plant species without and a high product without fertilizer and pesticide. We can get more production and healthy production of plant. Pesticide resistance, with the help of plant tissue culture, we can develop the pesticide resistance plant species. This is the in detail discussion. We have already discussed clonal propagation, production of identical copies of selected plants, production of pathogen-free plants, disease-free plants, propagation of sterile hybrids. Yes, one we missed, synthetic seed production. There are some plants have a hard seed coat, or their embryo just not grow normally. So there is a fail in the seed germination. In that case, we can prepare synthetic seeds with the help of plant tissue culture. Early we see the somatic embryogenesis. The somatic cells or the embryo formed through somatic cells further coated in the sodium alginate and prepared synthetic seed. We can see in the image, these synthetic seeds work as a seed and grow into new plant. So through plant tissue culture, we can prepare the synthetic seed of plants having a lower germination rate and lower embryo health. In forestry, it will also help. We will we firstly discussed the plantation crops. In the forest or in the field where the plantation is low in the forest and prepare forest area and rescue the forest area in this generation. Propagation of rare plant we already discussed. Shortening of breeding cycle. If we go for the conventional method of plant hybridization, it will take 8 to 10 years to prepare a plant with desired characters. So through the plant tissue culture and genetic engineering, we can shortening this breeding cycle and have a breeding early. Production of plant from seeds that otherwise have a low chance of germinating. Some plants such as orchids and rare plants which have very low chances of germination. So in this case, we can produce plant from the tissue culture technique. Now, limitations of the plant tissue culture. There is a contamination risk. If we don't, we will fail to maintain the aseptic condition, or somehow the microbe touch the culture, it will the rapid spread of contamination and the whole protocol will fail and whole experiment will fail. So if we or if there is a contamination, slight contamination, there is a widespread loss of cells, equipment, chemicals, etc. Risk of mutation arising. As we discussed, this explant or in tissue culture, there is a pamper child. One thing wrong, it will ruin the whole experiment. In artificial, we are trying to culture a pampered child with the help of artificial environment, which sometimes can induce the mutation in plant species. So sometimes the cells are not comfortable or not uh, cope up with the artificial environment and create the mutation. So there is a risk of mutation, and there is a response to tissue culture condition varies. There is a trial and error method to determine the optimum media or condition. Each plant have the specific condition for their development. Other one is specialized equipment required. If you want to prepare or build a tissue culture lab, there is so much specialized equipment required, such as laminar airflow cabinets, autoclave, water purification systems, glassware, culture tubes. There are large area having a washing area, and we discuss the whole plan. So there is a much to do and to have for the plant tissue culture. So there is a special requirement of uh, specialized equipment and higher cost equipments. The last or the higher labor cost. It is necessary to have a skilled labor for the plant tissue culture if we uh, introduce plant tissue culture in a commercialization. In commercialization, there is a um, higher need of skilled labor and higher labor cost required. So this is the plant tissue culture. I have gave only the overview. There is this is the sea, and we just reach to the shore and dip our foot in the water. So thank you for listening my lecture. I hope you found it interesting. Thank you.
बहुत अच्छा